So we are talking about high content information that ultimately takes account not just of the particular patient's disease state, but also of their genome and the context, which be their lifestyle, their diet, and so on. And again, just to emphasize it, it's all about small amounts of material here. So what we're talking about is taking labeled technologies and label-free technologies and expanding their footprint to be able to look at more cells or more proteins, or indeed drive the sensitivity down towards smaller numbers of cells. We'd also like added value, which I'm characterizing here in terms of length scale. You want chemical information about the degree of complexation. You'd like information about makeup of tissue. You'd also like to know what's going on at the chemical group level for post-translation modifications. So ideal world, you'd like to fill up this volume with the ability to deduce the information that you need. We focused on two types of platform. One is a fluorescently labeled platform, and one is a label-free platform. I'm going to describe a little bit about these now. The labeled platform is in some ways just an extension of a well-known antibody chip approach. That is to say, you have an antibody on a surface and you grab the analyte from solution which is labeled. The slight difference here is that we're doing this at the single molecule level. We're able to measure each individual molecule as it comes down to the surface, and this gives us essentially single cell sensitivity. And what you've got here is an antibody patch, with each cross being the location of a single molecule pulled out by a single molecule detection algorithm. Okay. This is a little bit of video of the molecules actually coming down onto the surface, so you can see this is what's going on. And in fact, although this looks complicated, it's quite straightforward to pull out the actual number of molecules being brought down on the surface, and this is what you see as a function of time. So it's straightforward to calibrate, and this we expect to be able to roll out for real use on real biological problems sometime in 2009. So this will allow us to actually study variation at the single cell level, the protein level. So this is not gene expression noise, let's call this protein variation. And it should be possible to multiplex 20, 40, maybe even 100 proteins simultaneously on one chip and to look at multiple samples on one chip. So that's, if you like, a slightly more conventional approach, but still an extension of the current capabilities. Something a little bit less obvious perhaps involves a label-free approach using spectroscopic and optical methods. Now, if you're doing spectroscopy on something as complex as a protein or any large biomolecule, the difficulty is that whether you're doing NMR spectroscopy or, for example, infrared spectroscopy, the spectra tend to be rather congested. There are so many features, you get essentially a blur, and you can't understand what's going on. Now, the NMR spectroscopists figured out in the 1970s that there are some very straightforward and powerful ways to overcome this. What you can do is, instead of exciting spins with your magnetic pulse, you excite a spin and then probe a short time later with another magnetic pulse to see if the energy has gone from one nuclear spin to another. And if the energy has, those two nuclei are coupled, and what you get is a cross peak, and if they're further apart, they're not coupled, and you don't get a cross peak. And what that does is it spreads this over-congested spectrum into two dimensions to give unique and distinct peaks, which gives you all sorts of information, not just analytic, but in case of NMR, the ability to solve structures. Now, I know I shouldn't say the word quantum mechanics in front of medics because it scares them. Nevertheless, the point is that once you understand level coupling as a generic phenomenon, you can think about inventing other spectroscopies that allow you to measure vibrational or electron coupling in the same way. So quantum mechanical coupling in level systems is a ubiquitous phenomenon, and I'm going to talk about a spectroscopy where we look at vibration couplings. One molecular vibration is coupled to another, and that's actually why heat flows in insulators. So the experiment is conceptually simple: an infrared pulse to excite one vibration, an infrared pulse to excite another, a visible probe beam to read out the information from that, and a detector to measure it, okay, with a sample in between. This is what the data looks like. This is one vibrational frequency here, this is another vibrational frequency here, and wherever there's a cross peak, it means that that vibration is coupled to another vibration. This is creatinine. If you're a bodybuilder, you have a lot of this in your blood, and it just shows that you get a nice information-rich pattern for a particular molecule. Now, it turns out, and people knew this but have tended to forget it, that you can identify a protein uniquely by its amino acid content. If you do a little bit of simple bioinformatics, you can recognize, for example, that if I know the composition of 4 amino acids in any protein to about 5% precision, it uniquely specifies 16,000 proteins out of a database of 33,000 or so. So what this means is that there is a form of top-down proteomics you can do, which allows you to identify the whole protein without breaking it up, as long as you can do a spectroscopy that allows you to count the number of these amino acids in a protein. And basically we're using this optical analog of 2D IR, 2D NMR, which is known as two-dimensional infrared spectroscopy, to do this. The proof of principle has now been demonstrated, actually just published a few weeks ago in PNAS. And the methodology has potential application in a whole range of these sort of omic-y type things, but also potentially in drug discovery and useful imaging, and of course one of our drivers towards high sensitivity and single cell analysis. This is what a workflow would look like. In many ways conventional, just replacing mass spectrometry with 2D IR. And what you can see here is a track of proteins read out by 2D IR. So this is a separation where the individual proteins are being read out in a track, and then they can be identified by the same method. You can image with it. This is a section of rat kidney. Okay, and what I'm going to show you is some differential contrast with the 2DR. We focused in on a hematoxylin dye peak, and you can see contrast, for example, between the blood clot and the surrounding tissue. The blood's got a lot of protein in it, and this is our first image taken with 2DR. Potentially has a resolution of half a micron. The resolution here is about 20 microns, I believe. And you can see here some differential contrast with the blood clot in the vein. Okay, so this is quite interesting because potentially you can do metabolic imaging and look for drug distributions in tissue. I have to say that's not been done yet, but it's on the way. In terms of post-translational modifications in chemical state, it's good to know how many proteins you've got, but you've got to know what form they're in because they really do have completely different functions depending on their post-translational modifications. And this just shows that we can pick up the difference between tyrosine and phosphatyrosine in a rather straightforward manner. And again, this is high sensitivity. You can do this at small amounts of material. And it's straightforwardly quantifiable. You know exactly how many molecules you've got in your sample in absolute terms. So the advantage of spectroscopy, it's intrinsically calibrated and doesn't have to go you don't have to go through any complicated methods. So where we're at is actually at a very early stage in the projects. The Mark I platforms, both labeled and label-free, are being rolled out, if you like, to use the language of instrument companies, in a lab demonstrator version in 2009. They have to be followed by Mark II platforms, which will have greater capability. And again, these are just lab demonstrators. They need rather specialized skill set to use, and they tend to be a little bit unreliable. What we're interested in doing now is exploring options for translating this into slightly more challenging bits of biomedical research, and we're looking for partners in all these areas, and there are lots of ways you can think about doing this. You can think about doing it through collaboration, where the lab demonstrators are adequate, and that's where the way a lot of our science, which is driving this, is going. We have people asking us about fee-for-service facilities. You can imagine spinning out the instrument, selling hardware or licensing, or developing a platform company perhaps which uses these tools for other things like drug discovery. So we're at a very early stage in these technologies. What I've described are not things that you're going to be able to buy this year or even next year, but perhaps within 5 years these will start to have an impact on basic research and will start to become available more widely to third parties by some of these mechanisms. And we're very interested to see and talk to anybody who is interested in helping us carry these forwards, because at the end of the day, the technologies have to be targeted appropriately at appropriate problems. Thank you very much.